Picture a first date. Somewhere reasonably nice. Not trying too hard, but not a fast food situation either. Two people are sitting across a table from each other, doing the thing that humans do on first dates, which is simultaneously trying to present the best version of themselves while also trying to figure out if the person opposite them is who they appear to be. And at some point, one of them looks into the other's eyes. Now, this happens on every first date. It is not unusual. But something specific is happening neurologically in that moment that most people do not think about. A rapid, largely unconscious extraction of information from the iris of the person sitting across the table. Not just attraction, not just connection, not just the romantic cliche of getting lost in someone's eyes. An actual information gathering process running on neural hardware that has been refined across millions of years of primate social evolution, attempting to read signals from a part of the human anatomy that turns out to carry considerably more information than the color alone would suggest. The eye color is part of it, but it is the beginning of the story, not the end. Because what eye color actually is, the biology of it, the genetics of it, the evolutionary story of why humans have the range of iris colors they do when virtually every other primate has dark brown eyes, is considerably more interesting than astrology columns about blue-eyed people being creative and brown-eyed people being trustworthy. And what eye color might actually signal, as opposed to what it is popularly believed to signal, involves genetics, evolutionary history, UV radiation, neurotransmitter chemistry, and the specific social dynamics of the human species in ways that make the simple version of this topic look like a cartoon of itself. Start with what eye color actually is at the biological level, because most people have a vaguer understanding of this than they realize, and the reality is more interesting than the usual explanation. The iris, the colored ring of tissue surrounding the pupil, is a muscle. Specifically, it is a flat ring of smooth muscle tissue whose primary function is regulating the size of the pupil by contracting and dilating in response to light levels. The iris controls how much light enters the eye for vision. The color that the iris displays is a secondary property, a consequence of the pigmentation of the iris tissue rather than a functional property of the muscle itself. The pigment responsible for iris color is melanin, the same pigment responsible for skin color, hair color, and the tanning response in skin. Melanin in the iris is produced by specialized cells called melanocytes, and the amount and distribution of melanin in the iris determines the apparent color. High concentrations of melanin in the front layer of the iris produce dark brown or black eyes. Lower concentrations produce lighter brown or hazel. Very low concentrations in the front layer, combined with the specific optical properties of the iris stroma, the fibrous tissue underneath, produce the appearance of blue, green, or gray eyes. Blue eyes do not contain blue pigment. This is one of the most widely cited surprising facts about eye color, and it remains true regardless of how many times it is explained. The blue appearance of a blue iris is a structural color phenomenon, the same type of optical effect that makes the sky appear blue. When light enters the iris stroma of a low melanin iris, the shorter wavelengths of visible light, the blue end of the spectrum, scatter more efficiently than longer wavelengths. The scattered blue light exits the iris surface towards the observer, making the iris appear blue even though there is no blue pigment present. The iris is essentially performing the same optical trick as the atmosphere. Green eyes involve a combination of low to moderate melanin in the front iris layer and a contribution from lipochrome pigments that add a yellowish tone which, combined with the blue scattering effect of low melanin, produces the perceived green color. The specific shade, from blue-green to yellow-green to olive, depends on the precise combination of melanin concentration and lipochrome content in that particular iris. Hazel eyes represent a combination of moderate melanin distribution that is uneven across the iris, creating the characteristic mix of brown, green, and gold tones that shift in appearance depending on lighting conditions and what colors are near the face. The heterogeneity of melanin distribution in hazel eyes is what produces the characteristic color variability that makes hazel eyes look different in different conditions. The genetics of eye color is where the story gets considerably more complex than the simple childhood biology lesson about brown being dominant over blue. For decades, eye color was taught as a textbook example of Mendelian genetics. Two brown-eyed parents cannot have a blue-eyed child. Blue eyes require two copies of the blue eye gene, and the inheritance follows clean, dominant recessive patterns. This teaching was convenient and wrong in interesting ways. Eye color is a polygenic trait, controlled by multiple genes simultaneously, not a single gene pair. Modern genomic analysis has identified at least 16 different genetic loci that significantly influence iris color, with hundreds of additional variants making smaller contributions. The two genes with the largest individual effects are OCA2 and HERC2, located on chromosome 15, but these are not the only relevant genes. TYRP1, SLC2, 4A, 4, IRF4, and several others also contribute meaningfully to the final pigmentation outcome. The result is that eye color inheritance is far more variable and unpredictable than the simple dominant recessive model suggests. Two blue-eyed parents can have a child with green or hazel eyes because the polygenic system allows combinations of minor gene variants to produce more melanin than either parent's eye color would suggest. Two brown-eyed parents can have a blue-eyed child if both carry recessive variants at the relevant loci that happen to combine in the offspring to produce low melanin expression. The inheritance probability distributions are complex enough that they effectively require genomic analysis rather than parental eye color inspection to predict with accuracy. This genetic complexity has an important implication. Eye color provides partial but incomplete information about ancestry and genetic heritage. High melanin expression is the ancestral human condition. Essentially, all humans had dark brown eyes until a mutation or series of mutations in the OCA2 and HERC2 region. Reduced melanin expression in the iris in a subset of a specific ancestral population. Eye color diversity, the existence of blue, green, hazel, and gray as distinct variants, is a relatively recent evolutionary development in the history of the human species. The question of when and where light eye colors originated has been substantially clarified by ancient DNA analysis over the last 15 years. Analysis of ancient European genomes spanning from the Paleolithic through the Neolithic and into the Bronze Age reveals that the genetic variants associated with blue eyes were present in European populations during the Mesolithic, the period before agriculture, but were not common. Early European farmers who arrived from Anatolia approximately 8 to 10,000 years ago, bringing the Neolithic agricultural package, had predominantly dark eyes despite living in Europe. The steppe pastoralist populations who expanded into Europe approximately 5,000 years ago, the Yamnaya and related groups, showed mixed eye color genetics. The frequency of blue eye genetic variants appears to have increased substantially in the European population after the Bronze Age through a combination of selection pressure and the demographic mixing of different ancestral populations with different genetic profiles. What selection pressure could have increased the frequency of reduced iris melanin in European populations is one of the more actively discussed questions in human evolutionary genomics. The most commonly discussed hypothesis involves the relationship between melanin, UV radiation, and vitamin D synthesis. Melanin in skin reduces UV penetration, which reduces vitamin D synthesis in sun-limited environments. Northern European latitudes during winter provide insufficient UV radiation for adequate vitamin D synthesis through skin, regardless of melanin content. But during the longer days of summer, reduced melanin increases the efficiency of vitamin D production. The hypothesis proposes that selection for reduced melanin in skin was adaptive in northern latitudes for vitamin D metabolism reasons, and that reduced iris. Melanin was either co-selected alongside skin depigmentation or is a genetic side effect of the same variants that reduced skin melanin. The vitamin D hypothesis is plausible, but contested. The specific genetic variants that reduce iris melanin are not identical to the variants that reduce skin melanin. They are on different genes with somewhat different mechanisms. Some researchers argue that iris melanin reduction was directly selected for separate reasons specific to iris function, while others propose that the iris depigmentation is primarily a pleiotropic byproduct, an unselected side effect, of selection on adjacent genetic regions. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. A second hypothesis proposes that light eye color was sexually selected, that in the specific demographic and cultural contexts of certain ancient European populations, lighter eye colors were preferred in mate choice, causing the frequency of light eye color variants to increase faster than natural selection on survival-related functions alone would produce. Sexual selection for unusual visible traits is well documented in other species and has likely operated on human physical appearance in various ways. If light eye color was genuinely rare in an ancestral population and rarity itself was attractive, the frequency-dependent selection dynamic, then sexual selection could have amplified the frequency of light eye color variants substantially beyond what adaptive selection alone would have generated. The specific social signal that light eye colors might have conveyed in ancestral populations, and what they might still convey in populations where they remain distinctively distributed, is where the biology of eye color intersects with the social psychology of human face perception. Human eyes are anatomically distinctive among primates in a way that has specific social signaling implications. The high contrast between the dark iris and the white sclera in human eyes, the visible whites, makes gaze direction dramatically easier to read than in other primates, whose sclera are typically darker and whose gaze direction is therefore less visible to social partners. The conspicuousness of human gaze direction has been interpreted as an adaptation for cooperative communication. Making it easy for others to determine where you are looking facilitates joint attention, and the kind of social coordination that human cooperative behavior requires. Iris color modulates this gaze direction signal in ways that have measurable effects on perceived attractiveness, perceived trustworthiness, and perceived arousal state. Research on pupil dilation, which is psychologically and socially significant as a signal of interest, arousal, and emotional engagement, finds that pupil dilation is more visible and more easily detected against a light iris than against a dark iris. Blue and green irises provide higher contrast backgrounds against which pupil size changes are more detectable, potentially making the pupil dilation signals of emotional engagement more readable in individuals with lighter eye colors. If pupil dilation is a meaningful social signal, and there is evidence that it is, with dilated pupils rated as more attractive in both sexes, and with the detection of pupil dilation associated with social bonding responses, then the detectability of pupil signals would vary with iris color in ways that have social consequences. Light-eyed individuals would provide more readable pupil signals to social partners, potentially making emotional engagement more transparently legible in their eyes than in dark-eyed individuals. Whether this detectability difference was socially significant in ancestral contexts, and whether it contributed to any sexual selection pressure on eye color, remains speculative, but is not implausible given the importance of pupil signaling in human social interaction. The relationship between eye color and pain sensitivity, personality, and health outcomes has been the subject of research that produces findings interesting enough to discuss and sufficiently preliminary to require appropriate skepticism. Several studies have found correlations between eye color and various physiological and behavioral measures that, if robust, would suggest that the genetic variants producing eye color are pleiotropic, affecting multiple biological systems, not just iris pigmentation. Melanin in the iris is produced by the same genetic pathways and the same melanocyte cell lineage that produces melanin in the skin, hair, and importantly, in neural tissue. The neuromelanin present in specific brain regions, the substantia nigra and locus coeruleus, both critical for dopaminergic and noradrenergic neurotransmitter systems, is related to the same melanin synthesis pathway. This biological connection between iris melanin production and neural melanin production is the mechanistic basis for hypotheses linking eye color to neurological and behavioral characteristics. Research has found correlations between darker eye color and higher pain tolerance in some studies, specifically dark-eyed individuals showing higher thresholds for certain types of pain and faster recovery from analgesic effects. The proposed mechanism involves neuromelanin's interaction with dopaminergic systems, with higher neuromelanin potentially modulating dopamine signaling in ways that affect pain processing. The effect sizes found in this research are modest, and the replication record is inconsistent enough that the finding should be treated as a hypothesis requiring further investigation rather than an established fact. Similarly, correlations between eye color and alcohol sensitivity have been proposed, with lighter-eyed individuals showing some evidence of higher alcohol consumption in population studies, and darker-eyed individuals showing some evidence of greater alcohol sensitivity. The proposed mechanism again involves melanin's interaction with neurotransmitter systems, specifically melanin's ability to bind and sequester certain compounds, including alcohol and related molecules. If neuromelanin in the brain sequestered alcohol-related compounds more efficiently in higher neuromelanin individuals, it could produce functional differences in alcohol metabolism and tolerance. These physiological correlation findings are genuinely interesting as research directions and appropriately uncertain as established facts. The point is not that eye color predicts pain tolerance or alcohol sensitivity with clinical reliability. It does not, and the effect sizes, even in positive studies, are far too small for any practical predictive application. The point is that the genetic variants affecting melanin production in the iris are part of a broader biological system that touches multiple physiological processes, and that understanding eye color as solely a pigmentation phenomenon misses the broader biological context within which iris color exists. The social perception of eye color, what people actually think and feel when they see different iris colors, is distinct from what eye color actually indicates biologically, and the gap between the two is where most of the popular mythology about eye color lives. Research on social perception of eye color has produced some findings worth noting. Studies measuring perceived trustworthiness of photographed faces have found that brown-eyed faces are on average rated as more trustworthy than blue-eyed faces by naive observers, but that this effect disappears when facial structure is controlled for. It is not the eye color per se that produces the trustworthiness rating, but the facial structure that tends to co-occur with brown eyes in the study populations. The face shape associated with brown eyes in the relevant populations may carry different dominance and trustworthiness signals than the face shape associated with blue eyes, and observers are responding to the facial structure rather than the eye color. This is a methodologically important finding because it illustrates how difficult it is to isolate the effect of eye color itself from the other variables that carry with it in specific study populations. In populations where blue eyes are common, the co-occurrence of blue eyes with specific facial features may differ from populations where blue eyes are rare, potentially producing different social perception findings across populations. The specific cultural meanings attached to different eye colors vary considerably across cultures and historical periods. In many East Asian contexts, dark brown eyes have traditionally been the standard against which other eye colors are measured and found unusual. The specific aesthetic attention to eye color in Western contexts, the high cultural salience of blue eyes in particular, reflected in everything from fairy tale descriptions of princesses to advertising for contact lenses, reflects the distribution of eye color variability in Western populations and the cultural history of how that variability has been aestheticized. In populations where all individuals have dark brown eyes, eye color variability is essentially absent and carries no social signal value relative to the reference population. The social significance of eye color, the social attention paid to it, the personality attributions made based on it, the attractiveness judgments modulated by it, is a product of living in populations where eye color varies meaningfully between individuals rather than a universal human perception phenomenon. The contact lens industry has created an interesting natural experiment in the social significance of eye color by making it cosmetically modifiable. People in many countries routinely wear colored contact lenses to change or enhance their iris color, with blue and green lenses particularly common in populations where these colors are less naturally prevalent. The popularity of iris color modification through lenses suggests that people believe, correctly or not, that eye color modification changes how they are perceived socially, which implies they believe eye color carries meaningful social signals. Whether colored contact lens use actually changes social perception of the wearer in the ways wearers expect is not well established by research. The interaction between artificial iris color and the natural facial features of the wearer, the visibility of the artificial color change to observers in natural lighting conditions, and the specific social contexts in which the wearer is observed all influence whether the lens color change produces the expected social effect. The anecdotal experience of contact lens wearers and the continuing commercial success of colored lenses both suggest that wearers find the experience valuable, but the objective social outcomes are harder to characterize. The question that the first date scenario at the beginning of this story was circling around, what does eye color tell us about the person across the table, has an answer that is simultaneously richer and more modest than either the astrology adjacent personality claims or the complete dismissal of eye color as meaningless variation would suggest. Eye color tells you something about the geographic ancestry of the person's recent ancestors, specifically about how much time their lineage spent in high UV or low UV environments over the last several thousand years, and about the specific population mixing events that shaped the genetic heritage they carry. In populations where eye color variability is meaningful, it provides some information about the specific genetic variants active in that individual's melanin synthesis pathways, which are part of broader biological systems touching neurotransmitter function and possibly various physiological response characteristics. It does not reliably tell you about personality. The associations between eye color and specific personality traits that circulate in popular culture, blue eyed people being this, brown eyed people being that, are not supported by robust empirical evidence and do not survive methodologically rigorous investigation. People perceive personality from eye color in consistent ways that reflect cultural expectations and the co occurrence of eye color with facial structural features rather than actual personality eye color associations. What it does tell you is something more fundamental than personality. It tells you something about evolutionary history, about population movements across continents, about the selective pressures that different environments imposed on different ancestral populations, all written into the iris of a person sitting across a table from you on a reasonably nice first date. Which is, when you think about it, a more interesting story than whether they are likely to be creative or trustworthy based on the color of their eyes. The biology is stranger and richer than the mythology. It usually is.